All right, in this video, we are going to look a little bit more in detail at the different types of cells within the nervous system and their structures and what they do, and we'll talk briefly about myelination and axon regeneration as well. So again, the two main cells of the nervous system are neurons, which are the ones responsible for the signaling, usually electrical signals, but chemicals also play a role. So they're responsible for those signals, either picking up sensation, so sensing that environment, or communicating some sort of response. And these are very large non-mitotic cells. So these cells do not undergo mitosis. So they are not making daughter cells. Glia, on the other hand, also known as neuroglia or just glial cells, these are thought to be mostly just support cells, and so their job is largely to help neurons do their jobs, essentially, and keep them healthy and stuff like that. So this is an actual real life view of artificially colored neuron cells and glia cells in the body. So this is what's called nervous tissue smear, and so you can see a neuron right here. This is that soma. We've got lots of dendrites. And then we're also going to have an axon. So this looks like an axon right here. We've got more branching out there. And then all of these glial cells surrounding that one single neuron. So we've talked a lot about these parts of a neuron already, so I'm not going to go into too much detail here, but remember we have that soma, we have dendrites, which are receiving information from other cells. And then the axon is carrying that information from the cell body. Neurons are only going to have one axon. That axon emerges from something called the axon hillock, and this is going to be covered in glial cells that form the myelin. And then the axon is going to end in some branches at what's called the axon terminal, and then the ends of those branches are going to enlarge a little bit to form what's called the synaptic end bulb. And that's really important for that signaling, because those end bulbs are going to meet up with the dendrites of another cell to pass that information on, pass on that signal. And then we also have what are called the nodes of Ranvier. These are gaps in between the myelin sections that are going to expose the axon a little bit. And so we're going to talk about why that's important here in a little bit. So here's that image. Again, we have our soma right here. We've got the dendrite projections coming off of it. You can see we're indicating another neuron up here with this end meeting up with those dendrites. All right, so dendrites again are receiving information. That soma will process that information and then send it down the axon to this axon terminal, and at the ends of each of these branches are the synaptic end bulbs, which are going to send that signal on to another neuron. And then along that axon, we have these myelin sheaths right here. So we've got the axon largely wrapped in that myelin, and then in between those bundles of myelin are the nodes of Ranvier. All right, so neurons can be classified a couple different ways depending on their structure. So neurons can have different structures. We have unipolar, bipolar, and multipolar neurons. So unipolar neurons, uni means one. These are only going to have that one process, which is axon and dendrite combined, forming from the soma. These are going to be found in the ganglia specifically. So those big areas like the synaptic ganglion. And then we have bipolar neurons. These are going to have two processes, one axon and one dendrite, emerging from the soma. These are found very commonly in the olfactory epithelium, so this is going to be important for our sense of smell, and then also in the retina, so sensing for vision. So we'll learn the retina later this week in lab when we cover the eye. And then multipolar neurons, this is a neuron that's going to have more than two processes emerging from the soma. And these are what's found everywhere else in the nervous system outside of the ganglia, olfactory epithelium, and retina. So these are going to be found in the central nervous system and some other ganglia as well. So this is what those look like. Here's those unipolar neurons. So our soma here, and then we're just going to have that one branch off, and so that'll be the axon into dendrites on one side, and then the axon terminal on the other side. Bipolar neurons, these are going to be branching off from two directions, not necessarily on opposite sides of the cell body, but just branching off in two directions. And then a multipolar neuron, this is more like what we've seen as an example of an actual neuron for the rest of this chapter that we've discussed so far. So we have these multiple dendrites branching off, and then this axon with the axon terminal. Neurons can also be classified and grouped based on where they're found, who discovered them, so what doctor or scientist found them, what is their job, what chemicals are they using to communicate with each other. So we have a lot of different ways of classifying neurons. We're not going to go into an immense amount of detail about all of them, but we're going to do a quick overview still. So we can have these pyramidal cells found in the cerebral cortex. We have these Purkinje cells, also of the cortex, but of the cerebellum rather than the cerebrum. These are those olfactory cells in the olfactory epithelium and olfactory bulbs, so they have special structures and stuff like that. But let's talk about functional classification. So what is their job? And this is very, very similar to how we functionally classify divisions of the nervous system with sensory, integration, and motor responses. So we have different neurons that do each of those. So sensory neurons, the dendrites of these are going to be picking up that sensory information, sometimes directly from the stimulus itself. So like sensory neurons that are picking up pressure, so some sort of touch, temperature changes, pain. Those are all sensory neurons. These can be any of those three main structures, physical structures that we talked about. Interneurons are going to carry out the integration part of the nervous system function, so retrieving, processing, and storing information, and they're also going to help facilitate communication between the sensory and motor neurons. Interneurons are only going to be found in the central nervous system, and they are always going to be multipolar, making up most of the neurons we have. And then motor neurons are the ones carrying commands out of the central nervous system to the various muscles and glands in our body, telling them to do some job. Produce this secretion, produce the sweat, produce the oil, produce the enzymes, move this way, whatever it is. So these are going to be found in the central nervous system or autonomic ganglia, and these are all going to be multipolar neurons. All right, so that's all about neurons. Let's talk about the glial cells. And we're going to start off talking about the glial cells found in the central nervous system. There's very similar ones found in the peripheral nervous system that are going to have similar jobs, but their name is different and their structure's a little bit different. But starting off here with the central nervous system, we have astrocytes, oligodendrocytes, microglia, and epidermal cells. So astrocytes, these are going to have tons of processes extending from their main cell body that are going to reach out and interact with neurons, blood vessels, and a bunch of the other connective tissues. So the meninges covering the central nervous system, these help make up the blood-brain barrier that helps prevent pathogens and stuff from getting into the central nervous system. That's very, very dangerous. Next up are oligodendrocytes. Their job is to help insulate the axons in the central nervous system. So a single oligodendrocyte cell is going to provide the myelin sheaths for multiple axon segments. So it's going to be working with multiple neurons. Microglia, these are some of the smaller glial cells, hence the micro part of their name. They are the macrophages found within the central nervous system. So this is our central nervous system's main immune response, is these macrophages. So they will help ingest and digest the cells that are dead, dying, breaking down, or any of the pathogens that do manage to cross that blood-brain barrier. And then the ependymal cells, they have a very, very specialized function. You will learn these as the choroid plexus in lab this week. Their job is to actually filter out blood to make cerebrospinal fluid, also known as CSF. And this fluid circulates through the central nervous system, through the ventricles, the central canal around the spinal cord, stuff like that. You'll learn the ventricles in lab this week also, but we have these special cells located within that choroid plexus that are filtering out that blood plasma to make the cerebrospinal fluid, because we still need to get nutrients into these cells within the central nervous system, but we can't cross that blood-brain barrier, so we can't actually get the cells directly in. We can't get those red blood cells directly in, but if we filter out the plasma to make the cerebrospinal fluid, then we can circulate that through the central nervous system to drop off those nutrients and pick up waste products, and then that will get back into the veins and return through the rest of the circulation to kind of get processed and stuff like that. So here's our graphic there. Here are astrocytes. So they're connected to a lot of different neurons. We've got the microglia right here. So little tiny macrophages to keep everything in good condition. Oligodendrocytes to make those myelin sheaths. And then here's that layer of ependymal cells to filter out the blood to make that cerebrospinal fluid. All right, moving on to the peripheral nervous system. We have two main support cells, satellite cells and Schwann cells. So satellite cells are going to surround the bodies of neurons and help support them. So just kind of general support and maintenance and stuff. They're found in both sensory and autonomic ganglia. And then Schwann cells are going to insulate the axons with myelin in the peripheral nervous system. So usually these are only going to wrap around a portion of one axon segment and no others. So this is a little bit different than those oligodendrocytes that are found in the central nervous system, making that myelin, and they're connected to those multiple axon segments. The Schwann cells, they're still making that myelin, but they're only focused on that one portion of an axon. So here's that graphic of our glial cells in the peripheral nervous system. So if this right here is a peripheral ganglion, so neuron cell body of a unipolar cell. Here's satellite cells, so just kind of basic support. And then a Schwann cell will find part of that axon, wrap itself around, and make the myelin around just that one section of the axon. All right, so we've talked a bit about myelin so far. So why is myelin even important? Why do we care about myelin so much? So axons can be either myelinated or unmyelinated. Unmyelinated is a slight misnomer. It's not fully unmyelinated. We'll get there in just a moment. But myelin is an insulating compound for the axons, provided again by the oligodendrocytes and Schwann cells, depending on whether it's the CNS or PNS. So again, myelin's very, very lipid rich, so very, very fatty tissue surrounding the axon, and this actually helps the transmission of the electrical signals along the axon. So having that insulation actually helps the electrical signals move along the axon better. So myelinated nerves are where those glial cells are wrapped around the axon with little to no cytoplasm between the glial cell layers. So segments of axon can be entirely covered by that myelin. Whereas unmyelinated nerves are axons that are only partially covered by myelin. So they're not fully unmyelinated and lacking that myelin altogether. They're just not fully wrapped around. So that myelin's not wrapped all the way around that axon. So like half of the axon will be covered, but not the other half. So here is that process of full myelination. So here is the axon in yellow. We get those support cells coming in like an oligodendrocyte. There's its nucleus, and it's going to get really close to that axon, and it's going to start producing that myelin, and it will keep producing it in this kind of sheet-like substance, or structure rather, and then it will start wrapping it around that axon very, very tightly so we get some multiple layers. And then we do that at multiple points along the axon with those nodes of Ranvier in between the different bundles of myelin. This is what unmyelinated axons would look like. So again, we still have that myelin present. It's just not wrapping all the way around the axon like it does in the myelinated axons. All right, we're going to wrap up this video talking about axon regeneration. So, can nerve cells repair themselves? And the answer is it depends on a lot of factors. And one of the big things that depends on is where those neurons are located. So in the peripheral nervous system, when nerves get damaged, the axons that are distal to the injury will degenerate. So they will break down. The ones, the piece, the broken off piece furthest away from that injury are going to break down. The Schwann cells that were wrapping around those injured axons are actually going to proliferate, so they'll make more copies of themselves, and they will form a brand new tube of that myelin that will help guide the regrowing axon from the proximal portion of that injured axon, so the part that's still connected into the rest of the nervous system. So those Schwann cells make that new tube of myelin that will help guide the regrowing axon through. They'll also provide some physical and chemical support. So most of the time in the peripheral nervous system, axons can regrow across the site of injury and grow back to their original targets. In the central nervous system, on the other hand, if a nerve gets damaged, the oligodendrocytes will respond to that injury by undergoing apoptosis, which is programmed cell death, and they'll go through that process or just in general become inactive. And this is going to lead to scar tissue forming in the central nervous system. When astrocytes sense that injury, so when we have those injury activated astrocytes, they actually release molecules that inhibit the growth of new axons. So they will actually release chemicals that prevent axons from regrowing. So these injured axons in the central nervous system are not going to be able to grow back out to their original target. And so nerve injuries within the central nervous system are permanent. So that's why if the spinal cord is injured, that is a permanent, lifelong injury for the rest of somebody's life, unfortunately. But in the case of a severe injury to like the arm or leg, where nerves are cut, or if somebody loses a finger, frequently, if it's done fast enough, that nerve, that axon can regenerate and regrow, and that person will regain full function of that finger that was cut off and then reattached. So here's our graphic of axon regeneration in the peripheral nervous system. So this is what we start with. So we've got our soma, we've got our axon with the Schwann cells around it making myelin. Here's our target. In this case, looks like muscle. We have an injury to that. Something breaks that axon. And so this section, again, the distal portion, degenerates. It breaks down. We've got macrophages out in our tissue in the body that come through, gobble all of that up, recycle it, get rid of it. And then the Schwann cells come in and start guiding that axon regeneration until it reaches out to that target site again. So pretty cool that at least part of our nervous system can repair itself. It's just not the really, really important part of it, arguably. It's all important, but central nervous system is pretty essential, and it cannot repair itself. All right, we will continue talking in the next video about communication between nerve cells.